Thank you for inviting me to be part of this course. So I work at the University of Bristol. I'm a genetic epidemiologist, so kind of work on the statistical side of epidemiology. So don't get involved in the lab. I couldn't tell you the last time I was in a lab, but yeah, do most of my work using statistical techniques on sort of high performance computers with lots and lots of data. So one of my main research interests is doing or looking at the genetics of eczema or atopic dermatitis. And so I've been involved in several large-scale genome-wide associations for eczema. And so what I'm going to talk to you about today is a lot of actually going through the process of doing that and trying to interpret the findings. And then we'll talk a little bit about some of the findings that have come out from that. I'm aware that I might be slightly covering some things you've already heard today, but as I say, I'm kind of coming at it from a little bit of a process side. And so I'm hoping that that will be useful for some of you that want some more insight into sort of how these studies work. So I'll go over our GWAS of eczema and talk a little bit about what we've found. Then I'm going to focus quite a bit on sort of how useful these SNP associations that we find in genome-wide associations are, and also talk about why they might not be useful or where their limitations are. And then following that, go on to talk about some of the bioinformatic follow-up that we often do to try and get a bit more understanding out of those associations. And I'll focus that on the kind of questions that we tend to address when doing bioinformatic follow-up, as well as the available data that we use and the type of approaches or statistical approaches that we might use to interrogate that data and combine it with the GWAS results. And then I'm going to also talk a little bit about why this sometimes might not work. And then finally go on to briefly talk about going beyond the bioinformatics. And although I'm calling them the next steps here, they're things that I guess you know I know you've just heard about from Gosha and maybe from other people in the course. So I'm going to show you sort of how far we can go with the available data and then what questions might still remain. So just to kind of bring us all onto the same page, so when we're doing genome-wide association studies, we're thinking about quite a specific type of genetic association. So here's a plot here with allele frequency along the bottom and penetrance of or effect size of that variant on the y-axis. And so you may have come across sort of Mendelian genetic diseases that are have influenced by SNPs or variants that are very rare, but have a really high penetrance. So kind of having that variant, you know, you're almost certainly likely to suffer from a particular condition. So with GWAS, it's important to note that we're really talking about sort of right up the other end of the scale and studying variants that are very common, so we can easily find them in the population, but are having much smaller effects. So on this scale, having low or modest effects. And so what we expect is that for a complex trait that is influenced by these kind of variants, there'll be probably hundreds or thousands of variants all having very small effect, making up somebody's genetic risk. And in terms of atopic dermatitis, we know that there's evidence for it being highly heritable and likely to be influenced by these kind of variants. And I've just put 2 bits of evidence up here on the slide. So both from family studies, one from a study that looked at your increased risk of eczema if one or both of your parents had the condition. And here we can see that you're over 3 times more likely to suffer from eczema if one or both of your parents have eczema. And similarly, twin studies have suggested that it's a really highly heritable condition with more than 80% heritability. So I just wanted to talk a little bit about what we mean about a GWAS association. And so, yeah, please interrupt with any questions you have here, because it's important that you understand this. This might be going over something that you've already talked about in the course, or you already know, but I just want to make sure we're all on the same page as to what a SNP association means in this context. So with a GWAS study, we tend to gather a selection of cases and a group of controls. And for a particular SNP, we're just comparing the frequency of a particular allele in the cases with the controls. So in this diagram I've put up here on the slide, if we look at the frequency of C alleles, if we look in the cases, 62% of the alleles within cases are C, whereas in the controls, 49% might be C. So there are certainly C alleles within the controls, and equally not every case might have a C allele, but hopefully you can appreciate there is this statistical difference between the cases and controls. So there's a much higher frequency of this C allele in cases, even though it's not completely deterministic for the disease. So what we expect is going on is that throughout the genome, there's going to be, as I say, hundreds or maybe thousands of these types of SNPs having very small effects between cases and controls. And if you're interested in the kind of statistics that we're doing in a GWAS, when we have a binary trait such as disease outcome, we're often doing a logistic regression. And so that's a regression where as the exposure we have our SNP, so that's usually coded as 0, 1, or 2 number of alleles. And the outcome is just a binary outcome, so cases or controls normally coded as 0 and 1. And we tend to include a few covariates such as age, sex, and markers of population ancestry, which can act as confounders in the association. And if you're interested in sort of what the regression equation might look like, you know, we're running a logistic regression equation with potentially quite a few covariates, but it's the regression coefficient for the SNP item in that equation that is what we're testing. And we want to know, you know, is there evidence that this beta coefficient is not equal to 1, basically. So is there any evidence for there being a slope, a relationship between exposure and outcome, or SNP and eczema in this case? So it's actually a very simple statistical test, and it's the kind of statistical test you see, you know, throughout epidemiology. So it's nothing complicated. I guess the complication comes from the fact that we don't just study one SNP or one exposure at a time. Because we're doing this in a genome-wide setting, we're just repeating this simple statistical test potentially 1 million times, one for each SNP, and we're getting one result per SNP as our outcome. So very simple statistical test. We're just repeating it many times to get a result per SNP. So in terms of what we've done with atopic dermatitis in the GWAS that I've been involved in, so the most recent published GWAS is from 2015. We're actually in the process of updating this at the moment, and yeah, hoping to have a much bigger sample size that we're going to report on soon, hopefully in the first half of next year. But the one that we previously published on has over 21,000 cases and over 95,000 controls from a number of studies around the world that have all imputed their genetic data to a common reference. So this 1,000 genomes is a reference panel that enables us to ensure that every study is examining the same set of variants. So our previous GWAS was predominantly European studies. We did have a Japanese study, RIKEN, representing East Asians, and there were some samples that came from the American studies that were of African ancestry or Latino ancestry, but predominantly this was a European analysis, as you know, is a bias that is quite common in GWAS studies. And each study, as I say, had common genetic data. They all carried out their own GWAS analysis, sent us the data, and then we were able to meta-analyze all of that together to come up with, for each SNP in the genome that was tested, a result that says how associated that SNP is with eczema. So here is kind of, I guess, the main summary of our results. So this is the classic Manhattan plot that I'm sure you've all heard about. So where every SNP is represented along the x-axis with its position in the genome, and then on the y-axis by how strongly it's associated with the trait. So essentially for every SNP that we've tested, we're getting a measure of how associated that SNP is with eczema. Everything above this dotted line meets a genome-wide significance threshold. So all of these peaks that you see in green are where we've found good evidence for association at those loci. So as Sarah was mentioning just before I started, the strongest hit that we see is absolutely on chromosome 1 around the filaggrin gene. I'm not going to talk about the filaggrin region in any more detail in this talk, but yeah, just to note that is the strongest signal that we see from a GWAS, even though that gene was originally identified through alternative methods. It's still the one that's coming up strongest in a GWAS study. And then a question was asked about the MHC complex, and yeah, absolutely, we see a signal within the HLA region on chromosome 6. So across the genome, we found 31 of these loci that met this significance threshold. And the ones that have those red arrows in this diagram are just the ones that were novel to the most recent study that we published. And so, yeah, I guess that's the summary that you often see in a GWAS study. The other summary that you see is a table of the top associations. So just to give you, this isn't all 31 of those, but just a snapshot of the top. So each locus you see represented by a single SNP, which was the strongest association at that locus. The nearby genes we've reported. And then you see the result for the association. So at this particular locus, it was the G allele that was associated with increased risk of eczema. And you can see that all these p-values are pretty low. So, you know, very good evidence for association. The other thing maybe worth noticing here is that, so some of these have been associated with eczema previously, but they'd also been associated with some other related conditions. So A corresponds to asthma here, IgE was a GWAS of IgE levels, and then there's allergic disease and hay fever and other kind of related conditions that you might expect to come up as having common loci with eczema. So that's kind of the headline results from the GWAS, and I just wanted to talk a little bit about, although we have found these loci associated, what haven't we found? And these, you know, GWAS are really designed to identify loci and not causal genes, which is maybe what we're more interested in, or not even to identify causal SNPs. So just to kind of clarify what I mean about that, here's a zoomed in diagram of sort of part of the Manhattan plot. So instead of seeing the sort of tall tower here of signals, you're seeing things a bit more spread out so that you can see within this locus where those SNPs are that are associated with eczema. And you can see that rather than there just being one single SNP that's associated, there's a whole bunch of SNPs that show sort of similar strong evidence for association. And the key here is telling us that lots of these are actually highly correlated SNPs. So they're SNPs that are in linkage disequilibrium. So they're likely to be on the same haplotype block and often inherited sort of all together. So there's lots of SNPs here that show association. And equally, if you go down to looking at the genes in the region, this region that shows strong association is spanning a number of genes, and actually there's not lots of genes kind of up and downstream of this that could equally be the causal genes. So to go from this association at a locus to understanding which genes actually might be impacted is quite a difficult step, and certainly not straightforward from just looking at GWAS statistics. Just to go over this idea of sort of SNPs as markers, so I've put a schematic here that just says if you imagine that you have a causal variant, so this lollipop here is a variant that we know to be causal for eczema. As I said, because it's correlated with SNPs around it, you actually see a handful of SNPs that have high correlation with this causal variant that would also be associated. And then as you move further away and the LD blocks start to break down, you find markers that aren't correlated with your causal variant and therefore aren't associated anymore. So if we were to look at associations here, we might say that, you know, these 5 in the middle are associated with our disease, and the sort of the 3 or the rest of the SNPs aren't. So because we don't know the causal variant, we're actually usually in a scenario where we have these 5, we don't know which of the 5 might be associated. And it's actually even worse than that, in that because not every SNP is captured in a genome-wide association, just the most common or just the ones that represent the majority of variation in a genome, the situation we actually might get is that the causal variant hasn't even been measured, and we're only capturing association at this locus because we happen to have these 2 that are correlated with the causal variant. So hopefully you can see that if this is the GWAS data you're working with, it's very hard to go from that to understanding, you know, where in that region the causal variant actually lies. So that's why we see this sort of quite complex pattern of lots of SNPs associated, and it's unclear which is likely to be the causal variant. Is that all clear, or does anyone want to ask any questions at this stage?
I think it's one of the clearest explanations I've ever heard of what you can find and not find from GWAS. So, you know, we always focus on these peaks and here's the result, but you know, that diagram you just showed us of the SNPs that might be missing and actually the causal SNPs, that is a very important point. Yeah. There have been a couple of questions on the chat, but I think we'll probably save those till the end. So if anybody's got any specific questions about kind of the methodologies or the explanation that we've had so far, let us know. Otherwise, I think probably continue, Lavinia.
Okay. All right, so that's kind of the bad news, if you like, of what GWAS can't tell us immediately off from the findings. But actually, you know, there's a lot that they can tell us. And so I just put a few examples here of what genetic marker associations can be useful for, even if you don't know the underlying causal variant. So one thing is prediction. And although for eczema and sort of many traits that I've worked on, we haven't quite got to a position yet that genetic variants are useful for prediction. For some diseases they have. So for coronary artery disease, for example, there's been hundreds of variants that have been identified as associated. And if you kind of add up your risk across every variant, if you take the people with the really highest genetic risk, that is actually starting to become quite an informative measure, and thought to be actually more informative than knowing someone's family history. So you don't need to know what the causal variant is, just the correlation is useful in prediction. So certainly once more markers are found, yeah, they can be used for prediction without knowing the underlying variants. And the second example I've put here is estimating genetic correlation between traits. Again, you don't need to know the causal variant, just seeing the common loci coming up for different traits is useful in this scenario. So I've pulled out an example here from one of the first ever studies to look at genetic correlation on a large scale, and they looked at smoking, genetic correlation of smoking with a whole load of other traits. And this heat map here shows whether these traits are positively in blue or negatively genetically correlated with smoking. And the one that came up strongest was actually a negative correlation with years of education. And I guess it's not surprising that smoking and education might be correlated, but perhaps it is surprising that they're genetically correlated, so that the relationship between those two might be have biological underpinnings. So you can do this kind of analysis again without knowing the causal variants, just knowing that the correlation between markers, between traits, can tell you something informative. And finally, something called enrichment analysis. So this is where you look at the overlap between your regions of the genome that you found to be associated, and seeing what other annotations those parts of the genome have. I'll go through in detail this on another slide, but just to say, you know, are your disease-associated regions also regions that are in open chromatin in a certain cell type, for example? So these kind of enrichment analyses across your GWAS results, as I say, can be useful even though you don't know the underlying causal variants. So there's 3 examples of how you can get biological understanding from GWAS without knowing what your causal genes or causal variants are. So I've just put up here a couple of interesting observations from the eczema GWAS that we did. So one is that some loci aren't associated in all ethnicities tested. So here's just one locus, I think this is the IL6R locus, where we see a really strong association peak within the European samples that we included in our analysis. And we see absolutely no evidence of a peak within the East Asian samples. The East Asian samples was a lot smaller than the sample size used for the European analysis, but nonetheless, we would have expected to see some sort of weak peak here if these variants were associated in that population. So that's interesting. Another thing that when we were looking at the genetic overlap or the genetic correlation with other diseases, we did actually find a lot of the loci that we identified as being associated had previously been associated with autoimmune disease. So as Sarah was saying at the beginning, although sort of not classically an autoimmune disease, actually there does seem to be quite a lot of biological overlap with autoimmune diseases, and particularly for inflammatory bowel disease, we saw quite a striking amount of overlap between the eczema variants and what had been previously identified for inflammatory bowel disease. And so finally, what we found when we did our enrichment analysis, so we did an enrichment analysis looking at something called DNase hypersensitivity. So this is looking at basically parts of the DNA that are hypersensitive, just means that they're open and able to be digested by the enzymes that are used in the test. And so it's basically identifying parts of the genome that are open chromatin, so they're unwound, they're ready to have transcription machinery attach and ready to express genes within that region. So it's kind of a marker of sort of poised parts of the genome that are likely to be functional in that cell type. So what we did, we looked at whether there was overlap between our loci and these open chromatin regions in a whole range of cell types, and basically plotted that enrichment out on this graph, and found quite a lot of enrichment for a wide number of immune cell types, actually, but specifically for Th17, which was quite interesting. Did someone want to come in there?
I think it was just a bit of background noise, I hope.
Okay.
Oh no, well, there is actually a question that Michaeli might want to ask live. So Michaeli, would you like to unmute?
I was wondering, the enrichment analysis you performed was only with the 38 loci you map with the Manhattan plot, or?
No, so we actually did it at a range of different p-values. So if you look on this plot here, we did enrichment analysis where the p-value met a p less than 10 to the minus 9 and less than 10 to the minus 6. So we actually used all of the data in the genome, and just for each threshold, p-value threshold, sort of calculated what the enrichment statistic was. So you can see when you just look at the genome-wide significant ones, that, you know, this is the enrichment you see here. So Th17's really come out on top. But, you know, we were seeing enrichment of immune cell types you're right back at where, you know, the p-value was less than 10 to the minus 3. So there's probably a lot of undiscovered loci in this region that are probably associated, but we didn't have power to detect them. And yeah, we see that enrichment across pretty much every p-value threshold that we tested. So yeah, that's quite an interesting point, actually.
I complete my question. If you have a underpowered analysis, you can still get information using the enrichment analysis you use, or?
So I missed the beginning of that question. Sorry. If you have what
if you have an underpowered GWAS analysis, so you don't see many peaks or you don't see any peaks in your analysis, can you get still information with the enrichment analysis, or?
Yeah, absolutely. I think you would. Obviously, the better powered your analysis, the more you will see these enrichment. The more you will see this evidence for enrichment. But you know, certainly this suggests that you would see those enrichments even with an underpowered GWAS. So I think that's, you know, something that some studies, if they haven't found many loci associated, might still go on to do some of these global things to see if they can get any biological understanding from their results, even without significant loci. So yeah, I think it's worth doing. Okay, all right, so I'll move on. So although I said you can't just take a GWAS results and immediately learn about the genes and the causal variants, that's obviously an aim of follow-up of GWAS studies. And really, you know, GWAS studies were heralded as a really useful source of data for drug discovery. So if we're identifying these new genes that are associated with a disease, they might offer new drug targets for pharmaceutical companies to focus on. And so we want to try and take this step of biological understanding, but how do we do it? So we think about kind of 4 questions, really. So can we identify the causal variant? Can we identify the causal gene? Which cell type is affected? And then taking it all, sort of bringing it all together and maybe taking it a step further, what is the mechanism or pathway that's disturbed? So they're kind of 4 questions that I'm going to go over what the processes might be to answer them. So in terms of the causal variant, there's lots of methods that aim to kind of fine map GWAS associations to identify either a single causal variant or, more commonly, something called a credible set, which is a small set of variants that you're you're quite sure the causal variant is likely to be sort of in that set. So you'll often see people report a 95% credible set where, you know, there's 95% probability that the causal variant is within that list that you're calling the credible set. And the bigger the GWAS, the smaller this credible set will get. So, you know, some GWAS have been successful in saying, actually, my credible set is size 1, so I've got 95% probability that, you know, this particular variant is the causal variant at this locus. More commonly, you see there's a handful of variants that fall into that credible set. There's a few different software that can be used to do this, and yeah, it's something that's worth doing to try and reduce down your numbers of likely causal variants. So once you've got this sort of smaller set of variants that you think might be or might include the causal variant, you kind of want to explore the functions of those. So are any of them coding variants? So do they disrupt the coding of proteins? Are there predictions that suggest that they might have sort of large functional consequences? So you can use the variant effect predictor tool to look at that. And then, as well as looking at predictions, you can explore sort of real experimental evidence that looks at whether variants might have a regulatory function. So we've already talked about DNA's hypersensitivity, but you can also look at other methylation marks from things like ENCODE and roadmap to say, is there any evidence that these variants are really likely to have an important regulatory function? You can explore this kind of data through browsers such as UCSC or Ensemble, and there are a couple of tools for specifically pulling out regulation functions such as HaploReg or RegulomeDB. And then in terms of the causal gene, I think, you know, we've just had a talk from Gosha that highlighted this, and, you know, I completely agree that eQTL data is really important to look at. And so here we're basically asking, are disease-associated SNPs also associated with expression of particular genes? There's some really good resources out there like GTEx and eQTL Gen for exploring this. And actually, there's now starting to be equivalent sort of protein QTL data sets that you can look at the same thing, but, you know, are your disease-associated SNPs also associated with levels of protein? So Gosha talked about co-localization, and so I was just going to say that, so as well as just knowing if your disease-associated SNP is associated with expression, you really need to then identify whether that is because those two associations are driven by the same underlying causal variant, or whether there's some sort of correlation or LD in the region that's causing that to happen. So the original co-localization paper is really good at explaining this, actually, and they basically show that with some sort of schematic diagrams that they're looking for a scenario where both the expression peak and the disease-associated peak are kind of exactly the same because they're driven by this underlying single causal variant. What's often seen is that though there might be two associations in the region, one with the eQTL and one with the expression and one with the disease, they might just happen to be in the same region and not actually driven by the same causal variant. So what this co-localization method is doing is really testing that this is the scenario that's happening and not this one. And the examples they give in the paper are really nice, actually, because here's a GWAS for LDL cholesterol. And when they looked up some results for the expression of a gene called FRK, they find that loads of SNPs in this region are also associated with expression of FRK. And so you'd think that that was really relevant and likely to be that this peak was driven by FRK as the causal gene. But when they actually do this co-localization method, they find actually 99% probability that it's this scenario for that particular locus. Whereas when they did a similar analysis for total cholesterol on chromosome 2, they find that this signal for total cholesterol and this signal for expression of SDC1 is actually 82% likely to be this scenario. So you can use this method to really work out whether these things that look like they're co-localizing really are truly statistically driven by the same variant. So another way of identifying the causal gene in the region is to look at chromatin interactions. So you might have a scenario where a functional variant is actually sort of genomically quite far away from the gene, but because the way that the chromatin kind of folds up and transcription factors might bring two bits of the genome together, in 3D space, you know, this variant here is actually very close to this gene and likely to have an enhancer type relationship with it. Whereas, you know, all the SNPs around here that might be sort of physically, genomically closer to the gene, actually in 3D space are further away. So, yeah, this 3D chromatin interaction data can really tell you about how SNPs that are further away might have regulatory influences on certain genes. Yeah, and just to say, there's data available online, and you can browse that for your particular cell types of interest. In terms of what cell type is affected, I talked earlier about how you can look at overall genome data to tell you something about important cell types, but you should also do this at individual loci. So most annotations I've talked about, whether that's gene expression or DNA's hypersensitivity or chromatin interaction, these are all really molecular phenotypes that exist in a cell, and they will differ from cell to cell. So you should always look at what is the cell type that you're interested in, and where you see some sort of association or some sort of data that's telling you that there's an annotation of interest, check that that's in a relevant cell type, because there might be some red herrings in the data that you're looking at. I said here at the bottom, but there's always an issue that it requires a cell type of interest to be available, and that's really a key challenge here, that, you know, some cell types and some cell states will have been tested and data reported, and there'll be others that are just missing. So whenever you're doing this type of thing, there's a whole load of data that you kind of don't know about, that you should be aware of as a limitation. Finally, in terms of this idea of thinking about the mechanism and the pathway that's disrupted, I think of this as sort of the local scale and the sort of much more global scale. So at a local scale, the mechanism I'm really thinking about is, you know, if a causal SNP, say, is in an enhancer, you know, does it disrupt transcription factor binding? What is that transcription factor? And sort of really having a good idea of how your SNP is having its functional consequence on the gene of interest. Because if you want to disrupt that or enhance it in some way, you kind of need to know what it is that you need to work with. So yeah, what's going on a local scale that's causing that functional relationship is really important. And then sort of on a much bigger scale is then how does that gene fit into a bigger pathway? So, you know, say here we think IL-6 is important, you know, IL-6 is part of a pathway. And so are the other genes in that pathway, are they also associated with eczema in the GWAS? And if you were to design a new drug, is there a good point or a bad point within that pathway to intervene? So just because a gene is associated doesn't necessarily mean it's a good drug target, and you might have to look elsewhere in the pathway to find the best drug targets. But there are good online tools for looking at pathways so that you can really build up a picture of how some of your different genes might be related in pathways. So this is just to reiterate some of the tools that I mentioned as I went along there. So yeah, there's some really good online browsers and ways to look up functional information. FUMA is a good tool for doing follow-up of GWAS studies. And another quite simple tool that's recently been released is the Open Targets Genetics tool, where you can put in a single SNP and it will tell you all of the evidence that relates that SNP to genes. So that's always worth looking at. None of these tools are kind of disease specific, so you'll get lots of stuff that's maybe irrelevant. So yeah, it's worth looking at how relevant the things that are coming up there might be for your disease of interest. And I guess, you know, echoing what Gosha was saying earlier, my personal opinion is if you only do one thing after GWAS, it's that comparing your GWAS results with EQTL and PQTL data using co-localization in relevant tissues, I think is, you know, a really great step in gaining that biological understanding. So just a few comments on why it might not work. So I've already said that these type of analyses are only as good as the data they use. So you really think, has the important cell type been studied? Has that cell type been under the right conditions or, you know, activation? It's possible that the effect you're interested in might only be seen in diseased samples, and a lot of the data that's available publicly is certainly from normal samples. So if it's skin samples, it's very unlikely to be taken from atopic skin. And also the sample sizes are often small, and certainly a lot smaller than the GWAS sample sizes. So if we really are trying to identify things that have tiny, tiny effects, then the very small sample sizes of some of these experiments might just be insufficient to detect what we're looking for. This has already been said by me and others, but yeah, just be aware of red herrings. Just because a GWAS region is annotated as an enhancer in an LCL, for example, it doesn't follow that that's automatically relevant for your disease. And so correlation is not causation. So one last problem, and I think this to some extent would be my answer to the question that Sarah asked Gosha earlier about what's one of the major limitations of something like co-localization. So I've said we're looking for this scenario where both your expression and your disease peaks are the same. So what we're assuming, I guess, in this case, is that the causal variant is affecting gene expression and that this is then causing the disease. So that's what we're trying to identify and what we're assuming when we see that co-localization. However, there could be some other scenarios. So the causal variant could be affecting gene expression, but also by some completely independent path could be affecting disease. Or the causal variant could by some path be affecting disease, and then just the disease state itself might be affecting gene expression. So just because you see these things co-localized doesn't mean that they're causally related. So this one refers to pleiotropy and this one refers to causation. And I just wanted to mention that there are methods to address these. So we would use something called Mendelian randomization to look at this question. And you can do something like looking at EQTL data in controls only to get around this second problem. So yeah, really thinking through what the plausible scenarios are and then trying to test them, I think is really important. All I want to say here is that, you know, this is all using publicly available data, doing bioinformatic analysis, and absolutely this needs to be followed up with carefully designed experiments to kind of confirm them. So though some studies might go as far as generating hypotheses or likely causal genes, unless they've been sort of causally validated in experiments, they are just hypotheses. I just want to say here that, so I've kind of focused on causal genes. I mentioned causal variants early on, but really I've concentrated on causal genes. And that's because if you care about identifying the gene because you care about drug targets, to some extent, you don't need to worry about the causal variant. But I just wanted to flag up here that if you wanted to do something like a single variant CRISPR experiment to confirm the causal gene, then you do need to know the causal gene because you need to edit the right variant. So it's kind of a means to an end problem. You only need to know the causal variant if it's the way that you're going to identify the causal gene, but yeah, it can be important to identify that so that you can do the right experiments. So I just wanted to finish on an example. So here is a locus that we identified in our GWAS, and as many of them, there's lots of SNPs associated with the disease overlying a number of genes. And so if we looked up the top SNP in some EQTL data, so this is in some GTEx data, we actually see that this SNP is associated with, I think, well, many genes. I think there's 7 different genes in this list here, and all in different tissue or cell types, you know, whole blood, lung, skeletal muscle. And even if we restricted it down to just skin cell types, then I think there's 4 different genes that show association in skin. So it can be really hard to unpick what's going on with some of these data sets. And we've just written a paper, and in fact, I'm hoping it's going to be out on bioRxiv either this week or next, as a way to kind of summarize all of the evidence. So for each gene in a GWAS locus region, we've tried to generate a score that summarizes all of the evidence for whether that gene might be causal or not. And you can see that for this particular locus, we get the highest score for PPP2R3C, and second highest for this CIAA0391 gene, and the rest of them are much lower. And when we go through and look at all the different evidence that has sort of contributed to those scores, we see a lot of evidence coming from co-localization or EQTL data sets, but we can also see a bit of evidence coming from chromatin interaction and maybe methylation QTLs and things. So although this is still sort of hypothesis generating, I think this is quite a useful way to summarize all of that evidence in one place to give people a sort of priority list of genes that might be causal in a particular region. I'll just finish now, but just to say that I have previously put a practical together to kind of try some of these tools out. So if you're interested, then please go to my GitHub page and you can follow some instructions for trying out, I think, 5 of the different tools that I've mentioned. And you can sort of think about what they might be telling you and also what they're not telling you, or what's missing, what other things would you need to do to come to some conclusions. I'll skip over these next 2 and just in the interest of time, but this just summarizes what you might find if you go and do some of those steps in that tutorial. But basically we find evidence that there's 95% probability of co-localization of a particular gene. This is CD207 in skin. So we think at that particular locus, it's quite likely that might be the causal gene. So just some take-home messages as my final slide then. There is a lot of useful publicly available information out there, but trying to interpret it properly is very difficult, and hopefully I've demonstrated that to some extent today. And if you're reading papers that do these kind of investigations, I guess I just wanted to highlight to you to remember the limitations and question the findings and think about what other analyses or experiments you would expect or want to see to really believe those findings. key things that I think about is the most relevant data even available? So can you look at the cell type you want to? And as I said, yeah, findings need to be validated in experiments to really get that confirmation that you found the variants or genes of interest. So yeah, that's it from me. So yeah, happy to take any additional questions that you might have.
Great, thanks, Lavinia. It's such a pleasure to hear you talk about GWAS and all these molecular genetic analyses, because you explain stuff very clearly. And even when you put up a scary equation on the screen, then you still explain it nicely to people that don't naturally speak equations. So thank you. We have got quite a few questions for you that have been coming in the chat, which is great. So I'm just going to choose a couple because we've already got 5 minutes now before the break. And any questions that we don't have time to answer now, Lavinia, I'll ask if you can answer them in the Slack channel.
Yeah, sure.
And that practical exercise that Lavinia just mentioned, again, I'll ask Lavinia to put links to that in the Slack channel, because it is we did that live on the course the last time we did it, and people enjoyed it. So if you've got time, I'd encourage you to give that a go. So it's hard to choose questions, but so Daryl had a good question. Well, they're all good questions, but I'll choose Daryl's to start with. He said, are we able to better identify causative variants by investigating patients with multiple clinically associated atopic diseases, i.e. patients with asthma, eczema and hay fever, compared to patients with just eczema? So I know that's work that you've specifically looked at, Lavinia.
Yeah, so I think that is useful, but I think they might be different variants or potentially a subset of the variants or loci. So, you know, when we have looked at diseases together in the past, and we tend to see that some of the variants that have been associated with eczema alone have come up and others don't. And the way I've interpreted that is that, you know, there are going to be some genes that are shared between all of these diseases, and then there's going to be some that are skin specific, you know. So I think the value that we've had from pulling all the diseases together is really through increasing the sample sizes. So if we can pull related diseases together and, you know, quadruple our sample size, then we just have more power because we have a better powered analysis. But I think that will probably only be interrogating a subset of those loci, because they have shared biology between them, and you'll miss the ones that are kind of specific to eczema or specific to asthma or the other diseases.
Yeah, I totally agree. And you've kind of shown that. I think it depends what question you're asking, doesn't it? Whether lumping all the diseases together increases the power or just muddies the water. But it's an important thing to think about, and it's something that we as clinicians can, you know, and should collaborate with basic scientists to kind of have this debate. So another question that's come up a couple of times relates to the numbers of cases and controls, and like the case and control ratio. So Michaeli said, if you have more controls over cases, does that improve your power to detect association? And Ali said, yeah, is there an optimum case to control ratio for these sort of big analyses?
So it's no different from any other epidemiological analysis, really. And I think the optimal case control ratio is 4 controls to 1 case. So lots of the studies that we use aren't actually specific case control studies, so they come from population cohorts. So it's not like people have gone out and collected cases and then gone out and found a sort of similar representative control group. Lots of these are population studies that have followed people for a number of years, and so within that we'll have a group of cases and group of controls, but haven't specifically selected participants at the outset into those categories or by those categories. So we've tended just to take whatever data people have available. For eczema, we're quite fortunate because it's quite a high prevalent disease that we get about 20% of those population those population cohorts will tend to have the disease. So we tend to end up with sort of quite nice ratios of cases controls naturally because of the prevalence of the disease. But yeah, you really want as many cases in your analysis as possible. And then I think you get more power as you get towards 4 times that sample size in the controls. And then beyond then, I don't think there's much additional power. But we've just taken whatever data is available. And because of the prevalence of eczema, we've ended up with, yeah, pretty nice sample sizes for both cases and controls.
Great, thanks. We've got 1 minute left, and I do want to ask this question because again, a couple of people have raised it, and it's about the ethnicity issue. So
yeah,
you know, you showed your map of the world, and it was a trans-ethnic analysis that you did, but yeah, notably African populations are really underrepresented in the GWAS that have been done so far. So are you aware of any plans? We have people on the conference this time from South Africa and other people that are very interested in including African ethnicity. So do you have, are you aware of plans for that to occur, and can you comment on why it might be important?
Yeah, so I mean, it historically is a big problem for GWAS studies, and they are, you know, the majority of studies you'll see are very biased towards European samples. Just to comment on like whether it's a problem or not. So I think it is a general problem of representation, and I think, you know, should be addressed from a representation point of view, if not from a scientific point of view. But from a scientific point of view, many loci, at least, maybe not individual variants, but many loci we see are shared between different ancestral groups. I highlighted one where we don't see that, but that was actually quite unusual. The majority of them we see shared loci. So the biological sort of knowledge that we gain from one ancestral population is actually relevant for many other groups as well. So I don't think it's sort of completely a problem for everything. You know, the fact that that diversity isn't there isn't definitely a problem, but it's going to be a problem for some loci. In terms of fixing it, I think it's tricky, just the way things have been funded. There just tends to be a lot more, you know, a lot of these big GWAS studies have been funded in European countries, and so that's what we've ended up with. There are definitely now some efforts in other countries and in other ancestral groups to get that data, but it's still really lagging behind. And also some of these diseases are much sort of less well characterized in other ancestral groups. So I think if anyone's aware of any data from some of these underrepresented groups, then we would love to include it. And, you know, we always try and include as diverse populations as we can, but we've really struggled to find the large enough samples with genetic data to do this. So yeah, it's a problem, and I think it's one that would be great to address. So yeah, if anyone does know of any.
That's a brilliant point to end, Lavinia. So yeah, anybody out there who's got data from Africa, get in touch.
I think Sarah's just frozen for a minute. The plan was to round up there. Lavinia, I guess the other thing I was going to add to that point was, apart from disease and control specimens in terms of SNPs in other populations, the issues with representation, I guess, link also in terms of how good the phenotyping is, and how good the genetic sample collection is, and also how good the reference sequence is, because even only 2 or 3 years ago, we're getting new normal reference sequence from different populations, and that made recategorizing of some disease-causing variants putatively to just less rare variants, just because more and more people have been included. So I think an interesting space to watch.
Yeah, definitely.
I think while Sarah reconnects, we should all just stop and thank you, Lavinia, for a great talk, and really enjoyed your session. We're going to leave the Zoom open now if people want to continue to chat, but this is meant to be a break otherwise. And then we are all due back at 12:30, which isn't very long, for our keynote from Fiona Watt. So we're looking forward to that. But with that, I'd like to close the session and thank you, Lavinia.
Thanks very much. It's been a pleasure.
Thanks for coming.